Okay, thank you for the introduction. I'm Felix Nwajei, one of the second-year neurology residents. Today, I'm going to be talking about a 74-year-old lady who came in. She has a past medical history of stage 4 metastatic melanoma. Presented 2 weeks after she started treatment with immune checkpoint inhibitors, specifically anti-PD-1 and LAG-3 antibodies. She came in with progressive droopy right eyelid, generalized fatigue, progressive weakness, and pain in her jaw and muscle. On examination, she had right eye ptosis, diplopia, tongue weakness. She has symmetric weakness in the upper extremities, 4+ out of 5, and also the lower extremities, 4 out of 5. Sensation was intact. She was intubated because she had a weakness of her respiratory muscles, unable to keep her airway open. We performed extensive tests, including muscle markers here. CPK was increased, aldolase was increased, hepatic enzymes were also elevated, and cardiac enzymes here, troponin, gradually elevated as well. Imaging of the brain using MRI did not show any structural lesions. Cardiac echocardiogram initially was normal, and over the next few days, gradually declined to 40%. Cardiac MRI was done, which showed regional wall motion abnormalities, and with a further decrease in the ejection fraction down to 30%. And we're concerned for immune checkpoint inhibitors associated myocarditis. And so, given the frequency of complications, neurologic, in this case, myasthenia gravis, myositis, or myopathy, and also myocarditis, we started treatment with a taper of methylprednisolone. Immediately, this taper was started, we started seeing some response. She was extubated. She still had the right eye ptosis. She had diplopia. Her strength slightly improved. She was 5 out of 5 in the uppers and 4+ in the lowers. The enzymes started decreasing. CPK, aldolase, AST, ALT, troponin all went down. We sent a myasthenia gravis panel, which came back negative. And then we repeated the cardiac MRI, which actually showed an improvement in the global and regional left ventricular dysfunction, and with the ejection fraction that went increased from like 30% to 71%. We performed EMG. We showed reduced CMAP amplitudes in the upper extremities, representing myopathy, motor axonopathy, or presynaptic neuromuscular junction disorder. Myasthenia seemed less likely because there was normal repetitive nerve stimulation, and she didn't have any reports of fibrillation or sharp waves. But talk about cancer immunotherapy complications and how to manage a situation like this. And so what you're seeing here to the left is just a depiction of what happens in the lymph node. You have an antigen-presenting cell, in this case, a dendritic cell or a macrophage, that presents a cancer-specific antigen to a T cell. And you have 3 signals that are needed for full T cell activation. The first is the antigen, which binds to the T cell receptor. The second is co-stimulation between CD28 on the T cell and CD80 or 86 on the antigen-presenting cell. Now, you can have upregulation of CTLA4, which outcompetes CD28 for the binding to CD80, and that can present a break to the T cell and prevent the complete activation and proliferation of the T cells. And you also need cytokines. So in the lab, when I was in graduate school, I used 2-photon microscopy to show the dynamics of immune response to the right. And there you see the tumor in blue. You have T cells in red. You have macrophages or dendritic cells in gray, and T regulatory T cells in green. That's to the upper right. And now to the bottom here, this is an enlarged view of a single cancer cell undergoing destruction, likely immune mediated. That's more extensive, but I'm just going to move forward from here. And so when you combine immunotherapy, in this case, anti-PD-1 and anti-LAG-3, PD-1 acts in the terminal stage of a T cell, and LAG-3 is upregulated in the tumor site. And so when you combine both of them and you compare the response to just nivolumab, that's just the anti-PD-1, you see that there's an increased median progression-free survival of patients over several months. And so you also have off-target infiltration of T cells to muscles. In this case, this study was published from Jim Allison's lab with T cell infiltration in mouse models and in the human myocardium. And here you have T cell in the tissue stained with CD3, macrophages F480, and regulatory T cells FOXP3. Now, this is a single center study also coming out from MD Anderson, where they looked at the complications in patients receiving immune checkpoint inhibitors. And you see that myasthenia gravis had the highest frequency, followed by myositis, and it goes down the list, myocarditis, and it continues. And the clinical presentation of in this patients included ptosis. Here you have about just 49 of those patients, dyspnea, limb weakness. This is just by frequency. And so our patient had some of these presentation findings. The diagnostic tools included antibodies like we tested for myasthenia, muscle enzyme, CPK troponin, a trophonium test, positive, ISPAC test positive, and electrodiagnostic tests, for example, EMG. And you can see that not all patients tested positive for any of this. So there's a variation in terms of how patients test and what you can see with like the diagnostic studies. The treatment options include corticosteroids, acetylcholinesterase antibodies, IVIG, plasmapheresis, and it just looks like the frequency of at least this is just a number of patients that received any of these treatments. Now, the outcome is also variable, with complete resolution in 12 out of 62, 34 showed some improvement, and 16 just deteriorated. And you can also have deaths in patients due to myasthenia gravis complications. In this case, you have about 23% of such patients. And when you combine myocarditis, the mortality increases up to 50 to 60 something percent. And it's in those patients who have myocarditis that usually would need intubation because their disease seems to be more severe. And so one method of actually trying to abrogate or mitigate such a side effect is trying to target the complications in a more mechanistic sense. And in this case, you go back to the drawing board and you look at the T cell interaction with the antigen presenting cell, and an antibody, abatacept or belatacept, which has been used in transplantation for several years, can be applied in this setting where it actually prevents the co-stimulation of a T cell. And so it prevents the binding of CD28 on the T cell to CD80 and CD86. And so it prevents the full activation of a T cell. Now, this was published in the New England Journal of Medicine in 2019. This is an example of a patient who presented with symptoms that were concerning for muscle-related myasthenia, myositis, and myocarditis, and had increased troponin, increased creatine kinase, and the patient had plasma exchange at about day 7 with a reduction in the nivolumab plasma concentration, and then was subsequently treated with abatacept at different time points with complete resolution of symptoms. So this is just moving things forward, at least in a more mechanistic sense, and I think a better understanding of how immune therapy and works and ways to inhibit the process will be useful in controlling complications. Thank you. No questions, okay.
Thanks, Felix. That was really impressive. So good morning, everybody. I'd like to introduce our guest speaker today. Our project last year on the D&I committee, the 2021-22 committee, our theme was community. And so our goal is to start increasing invitations to members of our community to come and speak. And we're going to start that today with our guest from our neighbor institution, NCCU, and that's Dr. Alex Marshall, who is an assistant professor of biological and biomedical sciences at North Carolina Central University. He's an adjunct assistant professor at UNC in the psychology and neuroscience department. He started his education at the University of Florida, where he got his bachelor's of science in zoology, and then followed that up with a PhD in pharmaceutical sciences at the University of Kentucky. Now he's at NCCU, where he teaches biological chemistry, neurobiology of drug abuse, introduction to biology, and biochemistry. He has more than 20 peer-reviewed publications, and his research which is a study of functional implications of GPCR signaling in aging astrocytes. Is funded by a grant from the Alzheimer's Association. And today he's actually going to speak to us on the neurobiology of alcohol addiction. So thank you very much, Dr. Marshall.
Great. Thanks so much for the invitation. It is nice to come and talk to doctors and hear all this immune stuff. I was like, I don't even have to do my introduction now. So thanks so much for all that immunology. I study the neuroimmune system. And so my goal today will be to discuss how the neuroimmune system changes with binge drinking. And so instead of giving a typical talk, I'm going to try to give an overview of my lab. So it'll be it might seem kind of abbreviated in terms of some of the topics, but my goal is just to give you an overview of some of the stuff that we're doing. And let's see, I know how to share screens. I do this all the time. Let's see if I can make this a little smaller. All right, there we go. All right, so just an outline, basic background about alcohol abuse, and I'll go briefly through what we study within the neuroimmune system, but my lab is interested in different aspects of alcohol use disorders. So we are interested in the experimentation phase, so binge drinking and some of the problems associated with binge drinking, but we're also interested in the long-term effects. So what happens when someone chronically binge drinks? Technically, we're the most interested in neurodegeneration, but I'll show you some of that as we go through. So again, I don't have to talk to you guys about this, but alcohol use disorders is a diagnosable disease. What I will say is that studies say that medical doctors don't ask those questions as often as they should. So hopefully after today, it's something that you at least consider. Particularly what I'll show you, hopefully, is that there's a lot of environmental factors and differences among people that actually influence the negative aspects of alcohol use disorders. Anyway, so AUDs are associated with a loss of control and preoccupation with the drug. In particular, it's also thought to cause tolerance of withdrawal. And this depiction is just to show the disparity within who drinks, right? So there's about a third of individuals, I think this is all from the United States, that don't drink at all. So the top third, they don't drink at all. The middle third, we're probably not concerned about them. Maybe they're having 2 to 6 drinks a week. What is the most problematic is going to be the top decile. And so there are people that are drinking anywhere between 74 to 75 drinks per week. So about 5% of the United States population is associated with having an alcohol use disorder. So this is an older slide. I should have taken out the word addicted. So in general, we are trying to stop using that word because of its connotation and the stigma associated with it. So we try to say alcohol use disorder or someone suffering from an alcohol use disorder. As it relates to the financial burden, it costs the United States approximately $250 billion a year to treat problems or outcomes associated with alcohol use disorders. And that includes everything from car accidents to people missing work. Importantly, those who are diagnosed with an alcohol use disorder or recognize that they have an alcohol use disorder are less likely to seek treatment. So only about 1/5 of the people who have an alcohol use disorder decide to seek treatment. And even smaller are those who decide to use any kind of pharmacological intervention. And we know from data that the combination of behavioral therapy and medical interventions improves the likelihood of not relapsing. And so again, my lab is interested, sorry, I told you I was a walker. I'm gonna try to stay still for those that are on Zoom. So for those that are, my lab is interested in the progression. And again, I think I made a nope. My lab is interested in the progression of alcohol use disorders. So we're interested in experimentation in terms of binge drinking, but also the classical alcoholic or someone with an alcohol use disorder. Again, those are words that we try not to use anymore. So we do look at binge drinking, and this spiral is just set up to show how reinforcing it is. So someone that experiments with the drug and binge drinks is more likely to have negative side effects associated with the binge drinking. And I'll show you today some preclinical data that says that it happens much earlier than you would expect. But because of those neuroplastic changes, they're more likely to have withdrawal and anxiety. That withdrawal and anxiety leads to relapse or more alcohol consumption. And eventually you have a full-blown alcohol use disorder and or alcohol-induced neurodegeneration, which is my particular interest. So we're interested in the neuroimmune response. And so just as an overview of what the neuroimmune response is, again, not trying to belittle you guys, but I know that we all study the nervous system from different aspects. So there are two cell types that I'm mainly going to focus on, so microglia and astrocytes. Astrocytes are kind of the support cells, so to speak, and they're associated with glutamate regulation, the blood-brain barrier, et cetera. Microglia are going to be the classical macrophages of the CNS. And so we study both of them and we manipulate them in terms of understanding their responses within alcohol use disorders. I'm also going to talk about cytokines, which have already been introduced. And so for the purposes of today, I'm really going to just say pro versus anti-inflammatory, or that's my hope, instead of going through the laundry list of the numbers that we study. But the neuroimmune system is particularly interesting to me because of its dual nature in terms of being associated with behavior and biology. So classically, when we think of the neuroimmune response, we may think of protection and recovery. So there's some kind of trauma or some kind of event, and you have this neuroimmune response that helps you recover from that event. When it's prolonged, you get excessive neurodegeneration. But what has came out in like the 90s-ish is this idea of psychoneuroimmunology, that these cytokines and these immune markers can actually impact behavior. I'm going to skip this slide for time's sake. But when it comes to the neuroimmune response and alcohol use disorders, it's been shown in clinical studies that there is dysregulation in some of the classical markers of the neuroimmune response. And so what this is showing is your brain versus your brain on drugs. So we have a control. Let's see if I can use the pointer. That's why I walk, because I'm so used to pointing and just using my hand. We have a control versus alcohol patient, and there's an increase in the microglia. So IBA1 is a marker for microglial activation, and there's a clear distinction between the morphology in those cells in this clinical sample. Not only are the cells changed, but those cytokines and the receptors that are associated with the neuroimmune response have also been shown to be upregulated. And so this is just an older paper where they looked at MCP1 and TLR receptors or toll-like receptors, and showed that in clinical patients with an alcohol use disorder, there was an upregulation in cytokines, the toll-like receptors, and in the glial activation. So classically, the reason that people want to understand the neuroimmune response is that they thought it was directly associated with the neurodegeneration that's caused by alcohol use disorders. And so again, your brain versus your brain on drugs. And so while alcohol use disorders are not classically defined as a neurodegenerative disease, there are studies that indicate that it is as disruptive as more classically defined neurodegenerative disorders, including Alzheimer's. But as I alluded to earlier, the reason that I want to study the neuroimmune response is because it's associated with both the biological response in terms of neurodegeneration, but also some of the behavioral responses. And so we all recognize that the behavior is dictated by neurotransmission. And so the reason that I put this slide in here is that it's often assumed or classically defined neurotransmitters are kind of the gold standard for study, but we now recognize that these neurons have receptors that respond to neuroimmune signals. And most interestingly to me, the signals are going to mimic or be associated with some of the ethanol pharmacology. So you have an upregulation in something like IL-1 beta or IL-6, these pro-inflammatory cytokines that are associated with excitation. Alcohol is known to depress excitation, and in withdrawal, you get an excess of glutamate, you have this hyperexcitability. The problem with the cytokine response is that it doesn't resolve as quickly as it's more persistent than the glutamatergic responses. So even though the glutamate goes down, you still have this excess excitation associated with IL-1 signaling or pro-inflammatory signaling. So these, in particular, these cytokines are gonna go in the opposite direction of ethanol pharmacology. And knowing that, it indicates that if we can interrupt or intervene in this neuroimmune response, then maybe we can affect ethanol's pharmacology, which means we can affect alcohol consumption. So the current AUD treatments, and I always get yelled at when I put this up. The last time I talked to a clinician, they were like, please put the FDA, like, you know, that these are the FDA approved ones and just stop saying AUD treatments. So these are technically the ones that are FDA approved. I know that other people give different drugs for alcohol use disorders, but all of them treat more of the behavioral aspects. What's interesting about the neuroimmune response is that we can disrupt behavior, but we also may be able to reverse some of the alcohol-induced brain damage. So both of them would be clinically relevant in terms of alcohol use disorders. So the brain regions of interest that I'm going to talk about today, I think, I only put these two, are the hippocampus and the amygdala. Our interest in the hippocampus, obviously, grossly, is associated with learning and memory. And so we are interested in both what happens during blackouts. So when somebody is actually intoxicated, how does the neuroimmune response respond? But also long-term damage to the hippocampus. It's one of the more susceptible regions to alcohol use disorders. We're also interested in the amygdala. The amygdala, again, grossly is more associated with emotional dysregulation. And we know that people that suffer from an alcohol use disorder are likely to have more anxiety or have more long-term emotional dysregulation. So the way that I did this is to place questions to help transition between some of the aspects of our study. So the first thing that I want to talk about today, and I'm going to check the chat just because it's a habit of mine. Okay. Is what is happening to the neuroimmune response during the transitionary phase. So what's happening during the binge-like consumption? And in order to do this, we use a rodent model called the drinking in the dark paradigm. And so the drinking in the dark, or the DID, as we call it, is a 4-day paradigm. It's great because the students can come in Monday through Thursday, and then they can actually go away for the weekend, which is not what I got to do in grad school. But they come in for 4 days. They give the animals access to ethanol. Animals, these mice are nocturnal. So the reason that it's in the dark is because they're more active. So 3 hours into the dark cycle, we give them access to ethanol for 2 to 4 days. So I'm sorry, 2 to 4 hours. Importantly, this paradigm does cause the animals or elicit the animals to drink to approximately 100, 140 milligrams per deciliter. Which is actually really hard in animal models. And so this is just pointing out that they're drinking not out of necessity, they're drinking because they are choosing, and I say choosing in quotes, to drink. And so what I'll show is just some characterization experiments in terms of what happens to the neuroimmune response. And so we do a couple of things. We're interested in the immediate response. And so you'll see there's some studies where we're looking at what happens at T0, so during intoxication. And then there's some studies that I'll show that are looking a week out, even a month out after these short binges of alcohol. We're also interested in when the transition happens. So we give 1 cycle, 3 cycles, 10 cycles of ethanol, and then we look to see when is this switch occurring? When is it more persistent? When is it more plastic? And so this is one of the studies that I did as a postdoc, where we were looking at some pro-inflammatory cytokines, and in particular, this one's on IL-1 beta. And what we were able to show is that specifically in the basolateral amygdala versus the central amygdala, we do get an increase in IL-1 beta, but it occurred statistically after 3 cycles. So yes, the 1 cycle is up, but stats are great. So after 3 cycles, it was statistically different. And this only occurred in the BLA. And so I'm not going to go too far into circuitry, but my lab is also interested in what regional spaces are more apt to be damaged or more apt to change. We also looked at, this one's not as pretty. We also looked at IL-10. So IL-1 beta, the pro-inflammatory cytokine, went down. Went up. IL-10, the anti-inflammatory cytokine, went down. So that's not surprising, but we did want to show that the effects are multifaceted. So we also showed the same thing looking at IL-10. And what this is showing is that, again, after 3 weeks in the basolateral amygdala, but not the central amygdala, you get a depression in that IL-10 response. It did seem to be specific to the CNS. And remember, this is like binge drinking, more like, more like you might do on the weekend, not calling you out, but it's not necessarily someone that is would be classically defined as having an alcohol use disorder. And so what we found is that it is changing in the CNS, but not the periphery. So that implies that the CNS is more susceptible to the ethanol effects than what we're seeing in terms of the immune response than what we're going to see in the peripheral system. We also looked at glial gene expression or changes in the glial cells. And so this work was done by a student at High Point University. And what she found is that there was an increase in microglial gene expression, but it seemed to resolve after the 1 cycle of ethanol exposure. We then went on to do this in a separate study where we did 3. And after 3 cycles, much like the cytokine change, the glial changes are more persistent. So this is Jay. He's a technician or a lab manager for us. And microglia have different morphology and they express different different proteins as they become more activated. Sorry, I'm so used to talking to undergrads. You guys are like, yeah, I understand what the CDU factors are. All right, so IBA1 is a marker for, it's in all microglia. CD11B is going to be more associated with the immune responding one. So instead of just surveying the environment, they actually have responded to something that's occurred. And so what we saw is that, yes, there was an increase in this gene, but we also saw that there was some kind of immunologic response by these microglia after ethanol consumption. And again, this is looking at different cycles. So both after 1 and 3 cycles of ethanol exposure, we saw this upregulation in OX42, and particularly at the 3 cycle, after 3 cycles. We're also interested in astrocytes. And so again, the astrocytic response mimics what we're finding with the cytokines and the microglia. And again, it seems to be a switch after 3 cycles. We've gone on to do 6 and 10 and still see about the same thing. So within my lab, again, gross overviews. So we're looking at the general response. And when I say general response, unfortunately, that means that we looked in male mice because that was what we did at first. Now there's been a shift in the NIH as well as in the scientific industry to consider sex differences more explicitly. And interestingly enough, there's a ton of studies, which is why, in hindsight, we're like, why did we only study male mice? There's a ton of studies that indicate that the female mice often have a more robust neuroimmune response than the males, and that changes with cycles. And so we've done some studies looking at sex differences, and maybe not surprisingly, we did see that the females had an even more robust response than the males to this ethanol. And so this work has been done by a student at High Point, Chris, and a new student at Central who's looking at the gene response. I also have an interest in aging. And so as Dr. Spector said, I have an Alzheimer's grant that I'm not going to talk about at all, but we've been looking at the differences in aging as it relates to alcohol. So there's, again, a lot of studies that suggest that alcohol might increase the susceptibility to develop Alzheimer's disease. And obviously that is associated with the neuroimmune response. Yes, sir?
You want to comment on that? I mean, as you know, that's controversial. Right?
Yeah.
What's your read of the literature?
So, the literature, I think, is more like, it's epidemiological studies, right? So it's hard to say that it's associated.
1 year is deadly.
Yeah.
15 a day.
So from my studies, I would say that there's so many things that can impact in terms of just exercise or life changes that I would assume that you would only see it with more chronic alcohol use. So I wouldn't subscribe to the one beer is going to make you more likely to develop Alzheimer's. But I do believe that those who have chronic alcohol use disorders are more likely to have, there's even alcohol-induced dementia, that they're more likely to develop Alzheimer's. And so we have some studies, which I'm not showing, that do indicate that it increases things like amyloid beta, aggregation. We haven't looked at tau yet, but we have looked at a bunch of the cytokines and glial cells.
The overall effects of moderate drinking do seem to be slightly bad rather than slightly good, correct? That's what I don't know about brain health, but that's what I read about the overall health.
Yeah, so not a clinician, but I will say that my thought is that the moderate drinking, again, if you're not intervening and you're also have a bad diet and you're not exercising, then yes. The joys of what I do is that I'm interested in the cellular aspect, but there's a ton of studies which hopefully counteract what I did back in the day that just suggest like exercising reverses all of this, right? So like literally there's a study that we did when I was in grad school where we put rats on a wheel and we showed that it reversed some of these glial effects. So, all right, so we're interested in aging. What's particularly interesting about what I'm showing is that most people think about the aging response or the pro-inflammatory side. What I'm really interested in is this dystrophic nature associated with long-term or aging. So you have this pro-inflammatory, I'm gonna cause damage, but then you also have the senescent, I don't respond appropriately and therefore also cause damage. And so what this is indicating is that, in my opinion, is that the aged mice, was this mice or rats? The aged mice have this ceiling effect in terms of my glial response is at its max. So even when there's this toxin, I'm not responding any further, and that could be just as damaging as having an excessive response. So I showed a lot of stuff just showing that the neuroimmune response happens. I then go back, so my lab says, hey, we saw this biological effect. Can we manipulate it to correct some of the issues that are normally associated with alcohol use disorders? And so I'm going to skip this slide for time, but there's a bunch of studies, clinical and preclinical, that suggest that immunomodulators, not neuroimmune, selfish, immunomodulators do have an impact on alcohol use disorders. Again, my interest is more in the regional specificity. So we do a lot of stereotactic surgeries where we will target either the basolateral amygdala, central amygdala, hippocampus. Obviously, what I showed you guys is that, hopefully you remember, that the BLA was more susceptible to the changes than the CEA. So in these studies, we targeted the BLA to interrupt some of those changes. And so the first thing that we did was we gave Kineret, or IL-1 receptor antagonist. And we did see that the IL-1 receptor antagonist, when directly targeted into the basolateral amygdala, reduced both binge ethanol consumption and associated blood ethanol concentrations. When we did the same thing in the central amygdala, where we didn't see the increase in IL-1 beta, it did not have the same response. So it maps on to the finding that where you see a response, if you can knock it down, it can reverse alcohol use as well. It can reverse binge drinking. So we also do a ton of behavioral controls. So if you see a decrease in consumption in mice, the assumption is that they're decreasing their consumption because of their desire, right? That's my goal, but I have to show it. So we also do things like locomotor tests, and then we look at other rewarding solutions. So when we gave them sucrose and we gave them IL-1 receptor antagonist, it did not disrupt their consumption of sucrose. It seemed to be specific to ethanol. It also didn't change their locomotor activities. So we looked at their time movement, both in the center and in the entire arena. The reason that we look at the center is because we also want to get a quick measure of anxiety-like behavior. So mice are more likely to stay on the edges. And if they're in the center, it's thought to be a symbol of some anxiolytic activity. So that is classical pharmacological interventions. My lab also does viral manipulations. So we do a lot of chemogenetics with the idea that we can disrupt certain cells. And are you guys familiar with DREADS already? Okay. So DREADs are a way of targeting specific cell types. So you can target dopaminergic cells, you can target CRF receptors. We're interested in the glial response. So we have both transgenic mice and viral vectors that are associated with GFAP. And we can manipulate the astrocytic G protein coupled receptor to either activate GQ or be a part of the GI signaling. And I don't say inhibit for astrocytes because it's not the same as neurons, right? So astrocytes are going to be much more responsive to the environment. And what we found, which I'm not going to show, sorry, I'm nerding out a little bit, is that GQ and GI actually have similar downstream signaling as it relates to the actual ethanol pharmacology. But again, I'm not showing all of that. I just try to grossly show that when we manipulate the astrocytes, and so this work was done by Drew, who's in dental school now. So when we manipulated the GQ signaling, specifically in astrocytes and the basal, sorry, this is grossly. When we manipulated it grossly in the transgenic mice, it reduced ethanol consumption. And so we have a reduction in ethanol consumption, specifically in those animals that had the GQ DREAD and the astrocytes. Unfortunately, it also impacted their locomotor activity. So what we found is that grossly inhibiting or grossly manipulating astrocytes or GFAP positive cells resulted in locomotor deficits that actually led to the decrease in consumption. But then we followed it up and looked specifically at the basolateral amygdala. So first we looked in these transgenic mice and we gave it peripherally CNO to activate the DREADs. And then we did the same study, but instead of doing it peripherally, we gave the CNO directly into the basolateral amygdala so that only the astrocytes in that region would be manipulated. When we did that, we again saw this decrease in ethanol consumption, but it was more specific to ethanol. So it didn't impact sucrose, and I don't think I have the locomotor activity, but it also didn't impact locomotor activity. And this work was done by the doctoral student in my lab, Kayla. So what we've seen is that there's a neuroimmune response, and my hope was to show you that it's independent of damage. So that 4-day model, the 3 times, even 6 times, there's not evidence of neurodegeneration. So this is just from binge drinking. You have these neuroimmune responses, and that when we alter the neuroimmune response, we can affect some of the behavioral implications of ethanol consumption. So for the joys of y'all, I also put in some of our classical pharmacologic interventions because you guys are like, I don't care what goes on in the circuitry, what happens peripherally that can affect general consumption. And so in this particular situation, I'm gonna kind of dance between the 2 models. So earlier I was only talking about alcohol consumption, binge drinking. For the pharmacological interventions, we've done both binge drinking and neurodegeneration. For the neurodegeneration models, you do have to gavage the animals. And so animals are much smarter than people. They don't drink to the excessive levels that cause neurodegeneration. And so we give them 5 grams per kilogram. There's a couple of models that we use, but at any rate, the blood ethanol concentrations are gonna be like 250 to 300 milligrams per deciliter. So much higher than the legal limit. So one of the interventions that we've done in terms of looking at this is etifoxine. So etifoxine, I got lazy. I didn't even spell out receptor. Etifoxine, as you guys may already know, is an anxiolytic and anticonvulsant drug that is commonly given. It's most classically recognized as being a GABA A receptor agonist. But there's new data that suggests that it's also a TSPO modulator. And so TSPO, while it is in the mitochondria, is more likely associated with glial cells. And so we believe that by modulating glial responses and by modulating GABA, you would particularly get a stronger response. So that's why we chose to use etifoxine in these models. So the first thing that we did was to look at our consumption model. And so this work was done by students at Central, Amaya, and Asia. And so what they were able to show is that when you give etifoxine peripherally, it does reduce ethanol consumption. And so this is looking at ethanol consumption versus sucrose consumption. However, I was a little disappointed, but such is life, that it didn't change alcohol-induced brain damage. So while we did see a slight reduction in the glial response, so we did look at microglia again, and etifoxine was able to reverse the alcohol-induced microglial activation, it did not necessarily correlate to changes in neurodegeneration. A part of it might be the dosing schedule that we chose. I think that we only gave etifoxine for 3 days after the ethanol consumption period. And so there's a lot of things about, maybe it would be better before or more chronically. But etifoxine has been associated with or thought to reduce neurodegeneration in other situations. So there's some studies out there about AD that suggest that etifoxine might be a good drug or good candidate for AD as well. We're also interested in environmental factors. So I already talked about our aging work. Tori did some stuff where we got money from the NIEHS to look at some of the adulterants that are often in alcohol, the most common being arsenic. And so we did the study where we showed that, if you also have arsenic exposure, you're more likely to have more damage or more glial activation specifically. We also do studies where we look at adolescent intermittent ethanol exposure. And so the purposes of these studies is that we give alcohol during adolescence, and then we look into adulthood. So they don't have any more access to alcohol. We just give it to them during this short period. And then like 30 days later, we look at the changes that persist. So the idea behind this consortium, I should say, is that we want to understand the persistent effects of adolescent alcohol abuse. And what we've shown quickly is that there are, again, these glial changes. And so this is Kayla's work too, where she looked at sex differences and found that alcohol does, again, increase GFAP immunoreactivity, particularly among females, because females have innately a lower number of GFAP positive cells in this area. So the reason that I put that in there is just to show you that in these models, in the various models, we're still getting this immune response. So what she did was to interrupt or try to manipulate it using donepezil. Donepezil is another AD treatment. It's an acetylcholinesterase inhibitor. While it's classically known as an acetylcholinesterase inhibitor, it's also thought that somehow, and this is where we just wave our hands, somehow it also disrupts some of the downstream signaling from toll-like receptors. And this is my favorite. I can't wait till I get to the point where I can put that in a diagram and it still be published, where I just say there's a receptor for donepezil. But so the donepezil receptor supposedly interrupts this toll-like receptor signaling. So again, dual actions on neuroimmune responses and may correct for the neurodegeneration. And so what she's been able to show, as well as our collaborator, Scott Swartzwelder, who's here at Duke, is that donepezil does reverse some of those alcohol-induced deficits. Particularly, this donepezil is given in adulthood. So they had access to alcohol in adolescence, and like 27 days later, I think we gave them donepezil. So the donepezil was able to reverse those long-term effects in both caspase-3, which is a marker for cell death, but also in the neurogenic niche. So alcohol can also reduce neurogenesis, and donepezil was able to reverse that as well. So my goal for today was just to show you that alcohol use disorders do cause a neuroimmune response. It occurs independent of damage, but it also occurs in periods of alcohol-related brain damage. So glial activation and cytokine responses seem to be a persistent thing within alcohol use disorders, and that disrupting the neuroimmune response has been shown preclinically and clinically to alter those consumatory behaviors, as well as not clinically for the damage, but preclinically for the damage, reverse some of the damage associated with alcohol use disorders. So the take-home message, because this is how I actually in my class with Cliff Notes, is that we looked at a variety of parameters and models, and I should change biological truth, but there seemed to be this robust biological change in the neuroimmune response. So whether it was males or females, whether it was aged or young, whether it was binge versus long-term, we saw this neuroimmune response, and that manipulating it impacted behavior and drinking. And so I'm at a point where I don't do the work as often as I would like anymore. And so this is just a quick acknowledgement of, I try to acknowledge as I go along of the people that did the work. So this is my lab. You might recognize this place. It is Thai Cafe. But the work that I showed was mainly done by those that were bolded. And we do have, grateful that we have so much funding. We have a ton of funding from NIEHS, Alzheimer's, NIAAA, NIGMS, and NIMH. And that is all that I'm gonna do. I tried to leave at least 5 minutes for questions.
Great.
So I'll be the chat monitor. Where are you going? Come.
As an aside, when you first handed me that pen, this is the difference, right? Like you guys saw people, I saw microglia. I was like, oh, look, there's little microglia.
Well, some people do say they're asked. That's fine. So anyone with questions, please enter them in the chat and we'll call on you. So the first question, how does alcohol get in the brain? Is it just diffuse across?
Yes, so alcohol is, oh gosh, I always get this word wrong, empathic. I always used to say empathetic, right? So it,
Empathetic.
it's empathetic. No, it's empathic. So it can, the old studies often used to say that alcohol just disrupts where water used to be. And that's why it's so promiscuous in terms of its effects. So yes, the short answer to your question is that alcohol is thought to directly cross the blood-brain barrier.
Okay. Any questions?
Sorry, I always like nerd out and then I'm like, nope, that's not what his question was. Yeah.
I'm just wondering, do you have a mouse model for alcohol withdrawal? And if so, do you ever try to manipulate the cytokines to see if it's short of withdrawal?
And repeat.
Oh, my bad. So the question was about alcohol withdrawal and whether cytokines have been shown to reduce that, or disrupting the cytokines have been shown to reduce that. We haven't directly done that in our lab. The individuals who did do stuff like that were looking at PDE4 inhibitors. So they were looking at gross changes in the immune response. And they did show that it shortened withdrawal periods. But those were preclinical studies. We actually do have a model where we look at withdrawal, but we haven't necessarily tried to disrupt it. We were trying to see when the most robust changes occur. Like, is it in withdrawal or is it afterwards? So we haven't looked at and disrupting it.
Do you see these same changes in the blood of mice, or is it just in the brain?
So the peripheral system during binge drinking, so early on, does not seem to be as impacted by ethanol consumption, but there are chronic studies that, and Fulton Cruz would say, he's probably somewhere like, Alex, make sure you say this, that the chronic effects on the liver directly impact the brain. So there's this whole idea that the immune response peripherally makes the brain more susceptible because of deterioration to the blood-brain barrier as well, to the peripheral immune response. And so that's one of the things that I kind of ignore because we're mainly concerned with the neuroimmune response. But we do collect the liver and we look at serum, and we have even collected like femurs to send people to look at the peripheral immune response too.
So I happen to think that there's a strong correlation between the metabolic changes of liver disease and Alzheimer's disease, because at its basis, Alzheimer's is a metabolic disorder. What about, I mean, you look at inflammation, but inflammation and metabolism are pretty tightly regulated. Have you looked at the metabolic effects in the brain or glia of this binge drinking?
So we haven't looked specifically at metabolism, which is funny because I teach biochemistry, right? But we are venturing in that aspect in terms of trying to understand some of the enzymatic changes that are occurring in astrocytes. But
But
mainly
it can just be like, you know, just some crazy metabolite. Metabolomics and inflammation apparently are like a hot thing as they interact with each other.
yeah, especially when it comes to alcohol. So one of the classical questions that I often get is, what about the acetaldehyde, right? And we don't necessarily look at that, but it is true that alcohol metabolism can also increase ROS production, which would also impact the neuroimmune system.
Or just
Which
alcohol's
haven't-
effects on regular metabolism.
Yeah.
You know, it doesn't even have to be the
pathological,
the
yeah.
alcohol, yeah.
But no, the short answer is we haven't directly looked at that, but it is an interesting, there's a lot of different avenues you could go in that, mainly because we're like a classical cell-based lab. We're interested in the ROS as it relates to metabolomics, but
The good thing about metabolomics is you don't have to have a point of view.
everything's metabolized.
All right, I'm still looking for outside questions. I'll give you one more, you know, unless someone else has one. Oh, go ahead, please.
I just wanted to ask you to say a bit more about the evidence in your preclinical model for neurodegeneration in these various protocols. It seemed like you were showing us increased IBA1 staining as a proxy for neurodegeneration, but have you actually looked for direct evidence of neurodegeneration?
Yeah, so I didn't show that. We use something called Florjate C, and Florjate B and C are more classically associated with cells that are actively dying, but we also look at things like caspase 3 and tunnel, and you do see evidence of positive neurodegeneration, so to speak, after these models. Particularly, again, that's in the gavage models. When the animals binge drink themselves, we haven't done it chronically enough to see like if years, it's also expensive, to see if years of ethanol consumption where they self-consume leads to neurodegeneration. But when we gavage them or we use vapor to force feed them high levels, we do see evidence of classic neurodegeneration. They've actually even done like the neuron loss and done MRIs to show the damage that are associated with these models.
One last one from me, still waiting. We had a talk last week, a small one, but the point of the paper is that if you look at the difference between rodents and humans, many of the inflammatory responses that used to be astrocytes and macrophages in the mouse, in the human are done by endothelial cells. Are you astrocytic centric or there are more endothelial cells in the brain than any other cell type?
That's a great question. So we have done, the question was the different cells that can elicit these immune responses. So we have done some work with using co-labeling protocols, which suggests that at least in our hands, especially early on, that these responses seem to be mediated by the glial cells. We have not looked at the co-labeling and the damage or the brain damage studies to determine if it's more mediated by endothelial cells. But yeah, there's a whole lot of literature out there, including my own, that shows the direct differences between species. So even if you look at mice versus rats, mice have a more robust neuroimmune response than the rats, just innately. So the same thing is true when you translate it up to humans.
Is there any potential, I mean, for your work in animals to really impact, you still need a human correlate of what you're seeing in the animals? Is there a way to get something like that?
Yeah, so the reason that we have chosen to study it is because the studies indicate that the chronic alcohol use disorder patients do have these markers. We haven't done any back translational work. I actually just found out about a mechanism for that, but unfortunately it has to be internal institution. So I'm not connected to a med school, so I can't do it. But yeah, there's a couple of,
We are.
yeah, I've actually talked to Scott Sportswelder about it. There's a couple of people who have been looking at directly looking at clinical problems, so whatever you guys see, and then back translating it into the animal models within substance use disorders. I haven't seen a lot of it for alcohol use disorders. Mainly, they've been looking at polysubstance. So one of the things that I ignored is that people that consume alcohol have other life problems. They have metabolic issues, and so yeah, they don't exercise. So understanding what the human condition is more like gives our models more face validity. And so that's been the premise of what I would like to do, but again, I don't have the money for that one.
I'd like to see how you'd get past an IRB. We're gonna get somebody very drunk and then we're gonna have them sign informed consent and do a lumbar puncture. Do you think it would fly, Andy? I mean, that's one of the, you'd have to find something in the blood to correlate it, yeah. Before they review it. Yeah. All right, everyone. So again, thank you so much for coming and giving us such a great talk. Don't forget your super.
I know that's yours. That's ours? Yeah, that wasn't mine. I thought it was cool. That's what I was like, I've never seen one of these.
Who's stronger was this stronger? That's not mine. That's been here.
Oh.
It was.
It says somebody named.
No, but it's got an Apple.
Yeah.
A binder.
No. I thought it was really cool though. I was like, yeah.
Anyway, thank you so much. So this is nobody's? It has someone's name on it. Thanks, Joe. Great job. Apple tag on. Great job, young man.
Oh, thank
Sorry
you.
about the name butcher.
No problem.
You know, the toughest name in your group to pronounce is Christine. It
Christine.
looks like Devises.
Oh,
It actually
Dizil.
pronounced Jeeves.
Jewish.
Jeeves, yeah, it's I
Huh.
forget where it's from, but I put it into Google Translate and came out with Jeeves. But I didn't put yours into Google Translate. That's why I pictured it.
Oh, I see. Google Translate during the job.
You probably would. You probably.
Thank you very much.
See you. Felix, nice job.
Thank you.
They really do encourage drinking.
Yeah.
Six, yeah. But no, we're, yeah, we will get together. Please say hi to Dan.
Yeah, it's a whole entire. But yeah, we would love to see you guys. I really thought about you a lot over the summer because I feel like we do this once a year thing around that time. Just like,
I know, but that's not very good once a year.
I
So you're teaching about what?
3, 4.
I honestly didn't know that we had 3 rotating.
It's just that piece. It's just that piece.
Okay.
So that piece comes in.
Oh, because medicine you'd be here earlier instead of med. Okay, yeah, see, I didn't know that. All these years.
That's why I was
And I
like,
I pretend I'm an exercise for everybody. Okay, put it all together now.
I
Right.
hope hopefully we get some time to see each other.
Yeah.
soon. Okay, bye.
Yeah, we could invite the other